A+ലേക്കുള്ള വഴി ഇനി തനിയെ തുറക്കാം. Subscribe to the ENCLAPS channel. Ring the bell button and open success. It's that easy.
Welcome all to the wonders of the living world. First of all, let me ask you a question. What is genetic engineering? Genetic engineering is the direct manipulation and modification of an organism's gene using biotechnology, isn't it? Genetic engineering plays an important role in daily life. The process in genetic engineering has influenced various sectors of life such as gene therapy, forensic, genetically modified animals and crops. First, let's learn about gene therapy. Gene therapy is the method of the treatment in which genes that are responsible for the diseases are removed and normal functional genes are inserted into their place. This has triggered a great hope in the control of genetic diseases. Now Tara has a doubt. Let's hear what's her doubt.
How can the genes that are responsible for diseases be identified from among thousands of microscopic genes?
Well, that is a very good question. Have you thought about it? Cells are the basic building blocks of the living organisms. The human body is composed of trillions of cells. Within our cells, there are thousands of genes that provide the information, the production of certain proteins and enzymes that make muscles, bone, blood, which in turn support most of the body functions such as digestion, making energy and growth. Sometimes the whole or a part of the gene becomes defective or missing from birth, or a gene can change or mutate during adult life. Any of these variations can disrupt many proteins that are made, which contribute to health problems and diseases. Gene therapy can be used to modify the cells inside and outside of the body. When it's done inside the body, a doctor will inject a vector carrying the gene directly into the part of the body that are defective cells. Now let me explain it with a video.
Genes make us who we are. Whether we are tall or short, whether we have blue eyes or brown eyes. Genes hold the answer to all this. What if the part of the gene is defective or deleted? It can cause a disease called cancer. The solution to this is gene therapy. Scientists discovered that bad genes can be replaced with one that is healthy, or they can even be turned off to solve a problem. Now, how do they do it? Say scientists want to add healthy genes to the cell. They need an efficient way to do it. It is by the help of vectors or vehicles. It helps to deliver gene into the cell. Viruses are a kind of vector. Scientists modify a virus and remove its ability to cause a disease, and the modified virus can be used to deliver the healthy gene into the cell. Gene therapy can happen inside the body by injecting the vector into the body or in a culture medium or by transfusing them into the body.
I hope Tara is cleared with her doubt. Now let's move on with our topic. Have you heard of HGP? What does it stand for? Yes, it stands for Human Genome Project.
An organism's genome is the entire genetic material of that organism. The complete set of DNA contains all the information required by the organism to function. It mainly consists of 4 nucleotides or bases, namely A, T, G, C. A gene is a section of the DNA that codes for a particular protein. Now, what is a DNA? Human genome is made up of 3 billion base pairs with 20 to 2,500 genes with an average gene size of 3,000 base pairs. It means that a large part of our DNA does not code for any gene or protein. Hence, we call them as junk genes. The genome of every person is 99.9% similar to each other. That means to say that we differ from each other by just 0.1%. The genes of humans are 98% similar to chimpanzees and 75% similar to rats. The Human Genome Project was an enormous project collaborated by many scientists from different countries to make the sequences and bases of the human genome. It was started in 1990 and ended by 2003.
The technology known as gene mapping helped to identify the location of a gene in the DNA responsive for a particular trait. The complete genetic material present in an organism is called its genome. In human DNA, the majority of the genes, except the genes that code for protein, is non-functional. They are called junk genes. What are the relevance of Human Genome Project? The human genome has about 24,000 functional genes. A major share of the human gene includes the junk genes. There is about 0.2% difference in DNA among humans. About 200 genes in the human genome are identical to those in bacteria. Now see if you can answer these indicators. What is the significance of Human Genome Project? The human genome includes about 24,000 genes present in its 46 chromosomes. The secret of the human genome is revealed through a project known as Human Genome Project, which started in 1990 and entered in 2003 in various laboratories of the world. The gene mapping is the technology that helped us to identify the location of a gene in the DNA. What is the benefit of genetic mapping? Gene mapping is a technology by which we can locate a specific gene in a DNA responsible for a particular trait. That's all for the day. See you again with more knowledge on genes and their products soon. Till then, bye.
3 ലക്ഷത്തിലേറെ വരുന്ന ചോദ്യോത്തരങ്ങൾ ഏറ്റവും കുറഞ്ഞ ചിലവിൽ സ്വന്തമാക്കുവാൻ ഉടൻ തന്നെ ഡൗൺലോഡ് ചെയ്യൂ എൻക്ലാപ്സ് ദ ലേണിംഗ് ആപ്പ്.